So today I'm going to quickly, going through some of the research and clinical case we have. But this is a free format, basically. And so Roland and I will just get things warm up. So after we finish our presentation, anybody want to discuss topic? I know, Jonathan, we have a presentation. Anybody want to present something? Just jump in, And I think we can present for a few minutes, maybe a couple minutes for question sections. So let's get started. And so for the MEG, and this is the MEG 101. MEG is a functional imaging technology, different from the structural imaging you get from CT, MR. We're measuring the neuronal activity, with a 1 millisecond time resolution and a 2 to 3 millimeter spatial accuracy in the cortical level. So when neurons are firing, and there's action potential, which is happening in the white matter, we're insensitive to that kind of signal because there's signal cancellation. Instead, we're mainly sensitive to the dendrite, the post-synaptic potential and current. If you look at this tiny score, you zoom in, there's different layer of neurons, 4, 5, and 6. Those are big cell, like a pyramid, called a pyramidal cell. Those are the one, when they're firing synchronously, that generate measurable MEG signal. When neurons are firing, say when I'm talking, my motor cortex, auditory cortex, language network, all the neurons are working pretty hard. The generate neural current. Neural current will produce magnetic field, which can penetrate different layer of head tissue with no distortion. And by placing the MEG sensor outside the head, we can actually measure the neuronal activity non-invasively. And here's a sensor we have called SQUIZ, the MEG superconducting quantum interference device that based upon the discovery by Brian Josephson and with Nobel Prize in Physics. So we can take measurements anytime, of the brain activity. Here's the auditory response. You can see this look like electric current dipoles, produce the magnetic field like the right-hand rule. By solving the MEG source imaging inverse problem, we can map the activity. The hotspot is what MEG can provide. Because what type of neurons, what group of neurons at what location, what time are actually activated. And the background, which is the structural imaging get from MRI. So MEG not going to replace CT or MR, which is adding the functional information on top of the anatomical imaging you get from MRI in this case. So the hotspot is what the MEG source imaging provide. And this is a brand new MEG, we have, and so Dr. Roland Lee is the PI. I'm also heavily involved. We get NHS 10 grand, $2 million. We're adding a few $100,000. We replace our old scanner with a brand new one. This one, the good thing about this one, actually, you have a 100% helium recycling rate. We can recycle the liquid helium, with 100% rate. That's great. Before we have a refilled helium once, every week, because the sensor, the SQUIZ sensor, has to be superconducting, has to be only working in the 4 degree Kelvin, minus 270 degrees Celsius. So without a helium recycler, our operational cost is super high. Now we have the 100% helium recycler, and the MEG signal is extremely weak. It's about 1 billionth of the Earth's field. So to make the MEG signal measurable, we need to block out all the environment electric magnetic noise by placing the scanner inside this room called the magnetic shooting room. This is potentially the best shooting room in the whole world for MEG with 6-layer shooting. And this is the shooting room re-established in the QI, the first floor over there. The door itself, you can see it's 2 feet XYZ, so it's a 6-layer shooting. And the shooting factor at 0.1 hertz is approaching 70 dB. Is super, super good. With this one, we can actually measure the brain signal. We can not only see the cortical activity, but the subcortical signals with the shooting room. So, if you have some experiment, physics experiment, engineering environment, you need very, very required electric magnetic shooting, that's the place to go, And so here's the examples, when they shock the median nerve, and that's a human sensory network light up in the contralateral hemisphere, and in this case, it's the left hemisphere. And with millisecond temporal resolution, by solving the inverse problem, we can see the signal propagation, 20 millisecond, primary central cortex, 3B and 1, and then later on, go to the secondary central cortex, S2. On the right-hand side, you can see this what I'll call Bible, the principle of neuroscience, but kind of short. In the chapter talk about human, sensory network, you can see the signal propagate from thalamus directly propagate to Bowman area 3B, posterior side central sulcus, area 1, area 2, exactly what we see over here. And then later on, relay the signal to S2, which is like a half rings, and physically at the end of central sulcus, which is this signal. So before, MEG, people use animal research, open the skull, put, electrode over there, and animals, non-human primate monkeys and other, animals. But now you can do that non-invasively, leaving human being with millisecond temporal resolution and a 2 to 3 millimeter spatial resolution. Here's what MEG provide: good resolution in both time and space. And we use MEG for clinical and also for research. And here's an example, MEG can be used for localized interaction spikes. I let Dr. Roland Lee, cover this case.
Great. So yes, so I'm a physician and working with the Ming-Sheng and the MEG. We've been together for, since what, 1990s? Yeah, then, that's right, 1997, I think. Anyways, so I want to tell you about what MEG is for clinically for real patients. All right, so it's very good for research, as Ming-Sheng will show, to understand how the brain works. But for clinical, there's two indications that are well established that the insurance companies will pay and the doctors recognize its value. One is for epilepsy, and two is for presurgical mapping of eloquent cortex. The most common indication we do it for, 90% of our cases, is for epilepsy. And just so, if you're not in the medical field, epilepsy, as you know, patients have what's called seizures. Sometimes they can just space out, but a lot of times they have this jerking movement that you can't control. And so this is due to parts of the brain that are just basically going crazy. It's like having a lightning storm in your brain that it's just out of nowhere, it just starts spiking. A lot of times, probably a majority of the times, there's actually something, if you do an MRI, you can actually see where abnormal parts in the brain that correspond to this. But the definition of epilepsy really is it's an electromagnetic disturbance, because that's how the brain works, as Ming-Sheng talked about. So in the past, before MEG, we only had EEG, and you'd put the electrodes on the surface of the head. But the problem is, when the electrode, the electrical activity comes through, the skull distorts it. So you can't see exactly where it's coming from. So people with epilepsy will say, well, it's somewhere in the right side of your head, right frontal lobe, but you couldn't see where. Well, the good thing is, most of the time, you don't care, because about 70% of the time, you just take some pills. There's a lot of different families of pills. You take the pills and your seizures go away. Well, if that's the case, you're done. You don't need to know where it's coming from. But there's about 30% of the patients with epilepsy are not cured or managed well with the pills alone. Those are the ones that we see. We don't see the other ones that are already cured with pills. So then the question is, okay, the pills don't work. We want to stop it. A good way to stop it, we found from medical science, is to actually, if you can find out where the spikes are coming from, you can cut out that tissue. And when you cut out the tissue of leptogenic abnormal tissue, then many times the patient is cured or very much improved. So that's the value of MEG, because what happens is, when we're looking at these traces here, as you can see where that region outlined in white, that's a spike. That's an epoch in time where the patient is having the spike. And the good thing is, because the magnetic fields are not distorted by the skull. So as you know, magnetic fields are really only influenced by metal, but you don't have metal in your head or your skull. So the magnetic fields come out, and you can do an exact calculation of the propagation of magnetic fields in the head. Now, it's more complicated to say, okay, we have the measurements on all the sensors, where in the brains are coming from. But if you assume at any point in time there's only one point in space, then it's easy to solve that problem. And so what we do is, at this point in time, we have to do a calculation of all the sensors here with the signal at each one of these sensors. You can figure out where in the brain comes from. And so when you do that, you can see right here, it's at that point in time, that's where the spike is coming from. And so you just look for different spikes at different times, and you'll get a cluster. And so this is what would happen in this patient. So we see this is the right frontal lobe.
It's pointer.
Okay, that's great. the right frontal lobe is where most of the spikes are coming from right here. And there actually was no real anatomic abnormality in this particular case. But the surgeons said, okay, well, let's go and take this piece of tissue where the spikes are coming from, most of the spikes, because very few are on the other side. And when they did that, the patient actually got quite a bit of improvement. But over a time, they got a little bit worse, and after a while, it was a problem again. So they asked us to go back and do the MEG again. You can see 3 years later. And what we find is that the MEG spikes are pretty much the same locations as they were before. And what happened is when they took them, they took most of the spikes, but there were some in the back here that they didn't take. Okay, so you know, it's gratifying though that we actually found the source of the spikes, and what they're planning on doing is now they're planning to do another surgeon, just take the rest of these spikes, and hopefully that'll improve the patient. So that's just one illustration of how MEG is really helpful, that it can take somebody who's epilepsy, who has uncontrolled seizures, and that by localizing where the spikes are coming from, you can remove that bad tissue, and in most of the cases we do, the patients see significant improvement.
Thank you, Roland. And so MEG can also be used for clinical cases of a patient's brain tumor. And with a structural CT MR, you can see the boundary of tumor, but not the whole story. Before the neurosurgeon removed the tumor, he want to know whether the tumor, removing the tumor would damage any major functional of the brain. If the tumor very close to the area control the body movement, remove the tumor and might damage the primary motor network. Language is very, very important for human. If a tumor is nearby, a language area, neurosurgeon need to know. So MEG can provide additional information in the nearby major functional center called pre-surgical mapping. So here's some more examples. I'm gonna skip this one because time limit. But this is one example when we do a motor test. You can see this case, and the pink arrow point to the tumor, which the enhancement, you can see that the white one is the tumor core. And it's very close to the primary motor cortex control the body movement. So using MEG, the hotspot of MEG that indicate localization of primary motor cortex for finger lifting. So when they perform the movement, they want to avoid that part. That's a new approach. And looking at the primary motor cortex, in the past, the subject need to repeat the finger movement 200 times. It takes about 20 minutes. Some patients just cannot do that because the body movement tend to be weak or non-time locked. So in this paper we published, we actually project the MEG signal, 306 sensors, and towards a much smaller, a subspace expanded by the EMG, the muscle activity, which only two-dimensional, two electrode pairs here and there. So by doing that, we enhance the signal-noise ratio drastically, and we sometimes only need 1 minute for sure. Sometimes even 5 seconds finger movement, we can localize primary motor cortex. So it goes from 20 minutes to 5 seconds. That's a huge improvement for us for patient care. So primary motor cortex, one of them, for another one's language. Language is so important for us. One of the major language area called the Broca's area. That's the language production area. So in this case, and the subject goes through the picture naming one, crocodile, airplane, baby, and apple, try to name the objects, and that actually activated. If you look at this lower left corner over here, the two green area are the typical Broca's area for language production. But want to know exactly where the one is. This is the 10 patient tumor, the 10 patient with brain tumor. Some of the tumor is so large, you can see that over here. We localize the Broca's area, and this using the radiology convention, basically the left hemisphere and the right side of the image. So the first 8 subjects, the left hemisphere Broca's area actually light up for language. That's what Broca claimed over 100 years ago. Our humans speak with our left hemisphere. So the left Broca's area in charge of language production for right-handed subjects. The last 2 are lefties. The left-handed. Their brain language localization is quite different, in some cases. So the subject number 9 had to be bilateral. Subject number 10, the language production goes to the right hemisphere. So for right-handed subject, they still have 50% chance with their left hemisphere control the language, but the other 50% go to either bilateral, go to the right hemisphere. And so, but localize the language production for patient with brain tumor is very important in the test for clinical service. And also we can also localize, and this is actually another study with healthy controls, and the different brain rhythm. We in 1924, Hansberg is the first one discovered the brain rhythm using EEG, but he cannot localize that one. And across decades, people, based upon invasive research, patient with brain tumor, that open the scalp, electrogrid, they get some idea what the brain rhythm look like. But here we can do that non-invasively in human being. The alpha rhythm is the top one, and the beta, gamma, and so on. So that provide a nominal database, and voxel by voxel. When we have a patient come over here with different disorder, we'll talk about the mild traumatic brain injury. Next one, we can compare the abnormal signal with the nominal database we established over here on healthy control. So I'm going to quickly cover in a couple minutes, and some research on mild traumatic brain injury. Mild TBI is a major issue on veteran population, and also in general population with motor vehicle accidents, sport injury and fall, but the activity, and the sensitivity using the traditional neuroimaging tend to be quite low. Structural imaging CT/MR sensitivity about 5% or less, and so try to develop a better technique for MEG localization. We're targeting the 1 to 4 hertz, and the theory actually established in the 90s, based on animal research called the deafferentation. I'm going to skip, the details of there, and either physical damage, to the pathway or some neurochemical blockage. So the idea is, can we do that, much better than CT/MR for detecting mild TBI? When people with mild TBI, one of the signatures, they had to generate abnormal bursts of 1 to 4 hertz oscillation or delta wave. We call it local sleep, because every night when you fall asleep, you have delta wave sleep. And what the brain is doing during that, non-REM stage 3 and 4, called deep sleep, the signature is low frequency rhythm, 1 to 4 hertz. It's called delta wave sleep for a reason. And during that process of time, and the brain doing something actually wonderful to try to clear up those toxic protein that you develop in your daytime, and the beta amyloid is one of the very harmful, toxic protein. So the brain try to do, its job like a janitor, clean up those harmful substance from the brain. As signature, the brain generates delta wave. Same thing over here. When the brain is damaged, due to mild TBI, the brain tries to isolate the area from the rest part of the brain and do the repairment or healing mechanism. So that's why we see this delta wave over here can be used for making diagnosis of mild TBI. Here's the case. 3 group: control, blast MTBI, non-blast MTBI, and the y-axis is indexed based upon the delta wave measurement. If you make a threshold over there, the exciting part is the small overlap between the control and 2 TBI group. So with this threshold, we get sensitivity 86.1% for the blast MTBI and 83.3% and for the non-blast MTBI. Combine the 2 groups together, we are approaching 84.5% sensitivity, much better than the structural imaging, for mild TBI. Another name for that is concussion, and for concussion MTBI, the CT/MR sensitivity only about 5%. We're approaching 85%. And because our sensitivity is very high, we can do individual, analyze in the diagnosis. This morning you heard a talk from Dr. Ramesh Jain. He was promoting this individualized medicine. That's something we can provide because of high sensitivity. We can look at individual subjects, the abnormal delta wave generation, and this is 8, TBI, and was abnormal delta wave generation. But compare that data with our normal database. And we also see good correlations with the different symptoms, personality change, trouble concentration, affective liability, visual problem, and depression. And I want to show just maybe a couple more slides, and I'll stop. Another biomarker we get with MTBI is different from the one I was talking about. We move from low to high. And the previous one talked about delta wave, which was 1 to 4 hertz, low frequency. But there's another signature at the high frequency end, which is in the gamma band, 30 to 80 hertz. So the mechanism is different in our brain. If you look at the neurons, usually you can divide into two general type of neurons. The excitatory, the big one, this red one look like a pyramid. They are these excitatory, in the pyramidal cell. And on top of that, there's the inhibitory interneuron, the blue one over there, to provide modulation either in the input and output end. So those are kind of GABAergic interneuron. And they're very important, and when they're damaged, because they're small, they're very vulnerable. When damaged, there's a lack of inhibition to the big cell, the red one, the pyramidal cell. So if there's a little bit noise in the background, the big cells start to fire, that generate abnormal high frequency and gamma band activity, 30 to 80 hertz. That's something we're going to show you in the next slide. I skip this one. So here's the main findings we have. We look at two groups, and the healthy control and the veterans with combat-related MTBI. And it's showing yellow means the TBI group have higher gamma noise, 30 to 80 hertz, compared with the veteran with no injury. You can see a lot of burn area in the frontal area, inferior frontal, and the frontal pole, and multiple parietal regions, and including angular gyrate, supramarginal gyrate, to produce those signature and high frequency gamma. So that's another biomarker we get from MEG on MR-TBI. So we're working with a different group. So we're open for business. You can, if you want to set up something, if you need understand the brain functions with a high temporal resolution in millisecond and spatial accuracy 2 to 3 millimeters, please contact us. We can set up and some pilot data acquisition for you guys, and we can collaborate. So and I think I'm gonna stop here and skip this one. And that's another. Thank my collaborators. Any questions? Yes.
Thank you very much for this interesting talk. Obviously, it's way beyond my level of knowledge. Just really interesting. It looks, one of the slides, it showed that the doctor seemed to cut the wrong part of the brain. So one of the slides.
It's not the wrong part of the brain. It's just that didn't completely follow the MEG result. So in this case, right. Yeah. So the MEG show, Dr. Lee mentioned that MEG showed the part generate potential the epilepsy discharge, but for whatever reason, the neurosurgeon decide not to cut everything, but cut only part of that.
Why would they do that?
There's a different reason,
So I think it depends. There are many reasons. First of all, I misspoke. There actually is a lesion there. I guess this is a patient no brain injury. The green arrows point out, so it's in the region of injury. So that makes sense. Sometimes in the operating room, there's a lot of stuff that happens. Like he'll be operating, you always have an anesthesiologist to make sure the patient's blood pressure is okay and so forth. So sometimes, the surgeon's cutting and he's doing what he needs to do, and then the anesthesiologist says, you have to stop. There's a problem, his blood pressure's dropping. We can't continue now. I have to control his blood pressure, and we have to stop the surgery. So a lot of times it's something happens in the operating room that the patient didn't react well to the anesthesia or something, but it's a team. And so I've seen this happen where the surgeon says, look, I need more time to do this. The anesthesia said, no, it is not safe for the patient to keep operating now. You have to stop. Sometimes that just happens. They might have bad reaction to anesthesia or something else. It could be sometimes intraoperatively they may check certain vital signs, stuff like that, and something is going wrong. That can be it. Sometimes they make a mistake. They co-register the location, and there's an error in the co-registration. With the patient, and because a lot of these surgeries are done where they co-register the MRI with the patient's the scan with the patient's head. You can actually have the reference system of this MRI can be co-registered with the patient by touching landmarks on the skull and so forth. You can do that. Sometimes an error in that. In particular, this is done when the patient's skull is intact before surgery. When you open up the skull, you'll see there's something called brain shift. The brain drops away from the skull once you open it. Once the door, the cover And so that introduces a little bit of error in the co-registration between the MRI and what actually happens in the patient's head, because it shifts. So sometimes the shift, they try and take that into account, but sometimes the shift is a little bit more than they expected. And so they make a mistake that way. So there's different reasons, but I work surgery all the time, and there's a good reason. They didn't just, they have the data, but something happened. So either in the operating room they were forced to stop it after a certain amount of time, or else there might have been an error in the co-registration between the intact brain and skull and dura, and then that brain shift. So that's what happens. The longer you're in surgery, the more complications there are, more things go on. So things happen in there, but you have to trust the anesthesiologist.
Thank you. Really, thanks. The second question is regarding, I heard about Neuralink, the Elon Musk's company. They are doing something like I don't know, is this similar or totally different kind of things? Can you comment?
Yeah. So Neuralink is, is like a brain-computer interface, and basically, and they have this implant electrode. It's more invasive. They open the skull, have implant electrode, but the electrode after the implant might communicate to the outside world through wireless communication. So you don't see this. Well, at least they are aiming don't have this wires hanging around. Not like the more traditional and implant electrode. This is more like wireless. But still, you had to do the surgery, put an implant electrode on the cortex. When they are doing that, they are collecting the neural activity, and use that one. In I think one of the cases they have a patient with a spinal cord injury cannot move their body parts, but with brain signal they can actually, with machine learning and AI they can actually navigate the web. We have actually published a paper. I think Troy Boo is the Ramesh mentioned that in the morning. He's a PhD student. Now he graduated, get his PhD. He's doing some on the machine learning using non-invasive approach with MEG. So MEG is totally non-invasive. So what he was doing, when we work on the project, I suggest take something more challenging. In the past, people use EEG, electric encephalography. They can differentiate the hand motion from left hand, right hand, which in my opinion is important, but not very challenging, because the right hand movement go to my left hemisphere, my left hand movement go to my right hemisphere, deep hemisphere, right? It's not as challenging as the his PhD topic. So for his PhD thesis, I want to do something more challenging. I thought, let's try the rock, paper, scissors with the same hand. It's almost the same muscle group, just different subtle hand gestures. Well, to see the non-invasive approach like MEG can differentiate this very subtle hand gesture, same muscle group. He did that, and the paper is published. We got something approaching 90% accuracy on single trials. So yes, answer to your questions, there's a way based on MEG can potentially provide non-invasive brain-computer interface, and in this case for hand gesture, we're working with students, Jessica, Bonnie, see other people over here. We'll try to see whether the MEG can actually move the mouse left and right, up and down, because the basic movement for typing, you can move to virtual keyboard and press the button, right? So we're working on the task, see whether MEG can be a good non-invasive based brain-computer interface. So different from the Neuralink, that are more invasive, but they have a higher resolution because they put the electrode right at the neuron. We are looking at something outside the scalp and non-invasive with MEG, but that's potential benefit, a huge benefit, and not go through the surgery with the surgical implant.
Thank you very much.
Thank you. Question?
I had a question just about MEG and how potential disruptions or changes in patient state would affect the results of an MEG procedure. So have you ever thought, or has it ever been in practice to do MEG while a patient was sleeping and see what kinds of brain activity levels would appear during that period and compare it to their awake state, and if that's possible?
Oh yeah, so every single patient with epilepsy, they come here, they take a nap. So this recording we show over here, and the activity we see over here when patient fall asleep. So they need to be sleep deprived before coming to our side. The reason of that, because the interactive spike usually happening in the non-REM stage 1 or 2. And so we wanted them to be on their seizure control medication, but to increase the likelihood to generate this abnormal discharge, want them to be sleep deprived. They come to our side, they're very sleepy, they fall asleep right away. So this is the recording when patient fall asleep. And the rhythm when they fall asleep have different signature than when the subject awake. When subject awake, they tend to be more higher frequency activity. When they fall asleep, typically during the delta wave sleep, I mentioned that associated with metabolic clearance, and the low frequency component dominate the recording. But occasionally, they have this huge discharge, like thunderstorm in the brain. So that's the example of when recording when subject fall asleep. But we also have great interest in sleep, in the sleep research, and in the healthy subjects, different sleep cycles. But the general conclusion is when subject fall asleep, during the non-REM sleep, they tend to have a low frequency one. When they have a rapid eye movement, REM sleep, then the frequency of the brain rhythm increase. You see the eye movement, there's a vivid dream. During that time, the brain try to do the memory consolidation. But when they wake up, they have more high frequency components.
I see. Thank you.
Thank you. Any other questions? If not, I think we are open to any other speakers. Maybe start with Jonathan, and you can introduce yourself. And anybody have a interesting topic, welcome to share with us. I think that's open platform.
All right, my name is John Venezia. I'm from the VA hospital in Loma Linda, a little bit north of here. I'm going to pick up a little bit on Dr. Huang's discussion of mild traumatic brain injuries, this time talking about a different sequela, functional hearing difficulties in this population. And I do need to tell you that I'm a full-time employee of the federal government, so this presentation reflects my views and not those of the Department of Veterans Affairs. This is some of the folks in our laboratory. So for those who aren't familiar with VA research, we function much like a typical academic lab. We have students, we have postdocs, we have funding from federal agencies. Our particular lab focuses mostly on perception and perceptual neuroscience. And here's a list of some of our recent projects. Today, I'm going to focus on just one of these areas, which is the effects of blast exposure on auditory processing in veterans. So let me give you a little bit of background to help you kind of get grounded in this topic. So what is hearing loss? Clinically, when we talk about hearing loss, we're talking about a reduction in the ability to just detect very soft tones. This is measured using something called the pure tone audiogram. So you hear pure tones or beeps at different frequencies, and we are measuring how loud those tones need to be for you to just detect that they are present in a quiet environment, usually over headphones. By convention, we plot increasing hearing loss going down on the audiogram. Do I get a cursor? That's okay. Oh, we're there. So in red, this would be an example of a normal audiogram. This person can detect tones at very, very soft levels. In blue is an example of an audiogram in someone who has hearing loss. The tones need to be louder for them to detect their presence. I suppose we'll try to get sound here over HDMI. Is that possible up there in the tech room?
It might be your computer setting.
Where Where's my mouse now? Let's just see what comes out. Sorry. So if you had a lot of hearing loss, that is about how loud the tones would need to be to just detect them. So that's an example of something like you might hear on a typical clinical hearing test. You press a button whenever you hear that signal. But what is it that folks with hearing loss actually struggle to understand? I'm going to kind of simulate that for you. You'll hear a podcast talking about hummingbirds in a background of like a simulated crowded restaurant.
In a coordinated way. There's some species of hummingbirds that actually will, Doug Usher will show this, that will flap their wings and create a slapping sound with that.
Okay, so that's usually the kind of situation where folks actually struggle to hear. And hopefully you can see straightforwardly that the difference between the stimulus in the beeps and the real world is very, very different. It turns out, though, that performance on a standard clinical hearing test is a pretty good indicator of how well you can understand speech in background noise. That's plotted here on this graph. So moving right on the x-axis, you have more clinical hearing loss. Moving up on the y-axis, you have worse performance on a speech and noise test. And there's a pretty tight correlation between those things. But you may also notice that, in folks who have pretty good hearing, those are the open dots, there's quite a range of performance. And the same is true in folks who have a little bit of hearing loss in the black triangles. So there's got to be something else going on kind of governing this ability. And so that's led folks to hypothesize that there might be something like two kinds of hearing loss that originates in the ear, where you get loss of the outer hair cells in the cochlea. This we might call peripheral hearing loss or threshold hearing loss. And then a different type that originates in the brain, which we might call central hearing loss, suprathreshold hearing loss, or now a more modern term has come to be functional hearing difficulty. And Dr. Huang gave you some of the info already on mild TBIs in veterans due to blast exposure. The relevant point with respect to hearing is that a lot of these veterans have been reporting in the last couple decades to VA audiology clinics with hearing complaints, but when we test them on a standard clinical audiogram, the test results are normal. So what's going on there? Well, the kind of straightforward hypothesis is that neurotrauma from the mild brain injuries is affecting that central or brain component of auditory processing. So let me show you just a couple of brief findings that we've come up with in this population of veterans who are blast exposed but have clinically normal hearing. So in one study using diffusion tensor imaging, we looked at veterans with a range of cumulative historical blast exposure and then tested their performance on a clinical speech and noise test. And we found that with increasing degrees of historical blast exposure, folks tended to perform worse on that clinical test of speech and noise. And that's controlling very carefully for performance on the standard clinical hearing test, PTSD symptom severity, age, and things like that. And when we went to look for neurocorrelates in the white matter, in this case, using diffusion tensor imaging, we found that a bilateral region of white matter areas in the brain were correlated with both cumulative historical blast exposure and performance on a clinical speech and noise test, much like that simulated restaurant that I played for you earlier. And the white matter region that was detected was actually something called the auditory radiations, the pathway that brings signals up from subcortical to cortical auditory centers. So you're getting reduced efficiency of transmission into the cortical auditory system in this population with increasing historical blast exposure and TBI. We then did some functional MRI in the same patients. We had a speech and noise task, kind of a more experimentalized version of that restaurant simulation I described before, where we had two competing talkers, one that the listener was supposed to focus on, a female talker, and then a male distracting talker who they were supposed to ignore. And we used a combination of spectrotemporal receptive field modeling and unsupervised learning to separate the bold responses or neural responses to the two talkers. And we focused our analysis on activation to the male talker, who was the interfering talker. The idea being that background noise is what the patients struggle with, so what is the brain doing in response to that interfering signal that they're supposed to suppress? And we found that with increasing levels of historical blast exposure, there was increased activation of a frontoparietal network that's involved in listening effort or cognitive effort to try and compensate for the presence of background noise. We also found in younger patients that with increasing levels of blast exposure, they failed to activate a medial frontal control network that is activated by healthy younger patients. So the typical pattern is that younger folks will activate this medial frontal network, shown in B there, to kind of separate the two signals using executive control, selective attention. Older folks are not able to use this network as much. But the folks who are blast exposed and young look like the older folks who are not blast exposed. So they're failing to be able to engage this network. So we see these kind of two patterns as a marker of the cortical network consequences of that diminished auditory signal coming in that we found with diffusion tensor imaging. So what are we going to do about this now? Well, we have a clinical trial where what we're doing for this population, mirroring what a lot of clinicians are doing in VA, is providing low gain amplification with commercial hearing aids. But in our case, we're comparing two ways of fitting or programming those hearing aids. The standard way is to use that clinical hearing test I talked about before, the audiogram with the beeps, and based on the outcome, there's a set of algorithms that decides how the hearing aid should be programmed, what frequencies to amplify, and so forth. But we've partnered with a private sector partner, Naughty LLC, to implement a speech-based version of programming hearing aids. And much like what happens when you go to get fitted for eyeglasses, where you kind of try different lenses and prisms and you tell the optometrist what helps you see with better acuity, we are doing the same thing with hearing aids, where we simulate a hearing aid and allow the patient to listen to different amplification profiles and tell us which one they like best. And we use a little bit of machine learning under the hood to kind of efficiently find the right program. And so one of the outcome metrics that we'll have in this clinical trial is that same fMRI task, where we compare the neural activation in response to aided versus unaided listening with these two different methods of programming hearing aids, the standard and the speech-based one. And we hope to see a reversal of those kind of maladaptive patterns shown by the patients with blast exposure. One of the problems with hearing aid research inside the scanner is you can't actually take hearing aids in an MRI scanner. So in this case, our private sector partner, Naughty, who will have a booth this evening at the open house, so you can go see them, they've developed a real-time master hearing aid, or a software simulation, basically, of all the modules of a hearing aid, so that we can just run the hearing aid on a computer that pipes into headphones. Another problem is that the MRI scanner itself is kind of a giant loudspeaker, so it's really, really noisy inside the scanner. Not great for making kind of fine discriminations between two slightly different mild gain profiles. So to solve that, we've partnered with another private sector company, Optoacoustics, who has developed active noise canceling headphones for the MR environment, so that we can run the scanner just as we would normally, but attenuating the noise that the machine itself makes. And it's worth noting that both hearing aid processing and active noise cancellation introduce some latency in the digital signal processing. And that's not a great thing if you want to do, say, testing of a speech task in a realistic context, like maybe audiovisual. So one of the things we worked on with Optoacoustics is to install, they developed and we installed a zero latency version of this active noise canceling technology for MRI. That's what we'll be using in the clinical trial, and maybe I can get you a demo of it. You'll hear the scanner noise kind of going along, and then the ANC will turn on and crush down the scanner noise, and you'll hear someone talking while the noise cancellation is on. Just got to get my arrow back. That's the scanner noise. That's ANC on.
1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6.
So you can even hear whispering with pretty good clarity while that zero latency ANC is running. So that's what we're up to. And with that, I want to thank you and our funding sources. Any questions? And certainly we are interested in thinking about how we might adapt this to the MEG facility here.
Thank you. Thank you. It looks like, again, this question may be naive. Looks like the veteran just need a noise canceller headphone with some speaker inside the ear.
Yeah, so right now we are focusing on just prescription of amplification to see if tweaks to those recipes will make a difference. Well, yeah, in practice, modern hearing aids and just off-the-shelf, commercial audio equipment does have noise cancellation. That's pretty good. It tends to only be good at the lower frequencies, not so good at high frequencies, and tends to be not so good at filtering out unwanted background speech. That's always been a big problem. But I know that a lot of the commercial hearing aid companies are investing a lot of effort in deep learning-based noise cancellation algorithms that are just now starting to come out on the top of the line hearing aids. So, in clinical practice, it's standard procedure to turn that stuff on for the patients and let them get maximal benefit from it. How good it's going to work out in the real world, still unknown with these new noise canceling algorithms that are coming out.
Okay, sorry. The second question is, it seems your research is focused on the blast-exposed veterans. There are many other reasons that people have hearing loss. So, and would this research result be helpful to those other people as well? Or?
We are certainly carrying out parallel investigations of these speech-based hearing aid programming techniques for other sources of hearing loss. So the kind of peripheral hearing loss that I mentioned, originating in the ear, that causes all kinds of distortions to the incoming signal that the brain adapts to, sometimes in maladaptive ways. So there's reason to believe speech-based programming could be good for that too. It could be good for the kind of hearing deficits that come on in middle age that a lot of folks start to notice when the ear is still working pretty good, but the brain is starting to veer off a little bit. So we do hope that it will be kind of a catch-all that will add some benefit in all those situations, but again, empirical question, so we're definitely exploring that.
Thank you.
Yeah, Jonathan, excellent talk. And so in one of the slides you showed us, in fMRI result, and the young folks, with blast MTBI to behave like older population, and was signature as the medial frontal control network. So is this the reduction of signal in the area? Or?
It is. Yeah. So actually digging into the nuts and bolts, we use, again, unsupervised learning to partial out the response to the interfering talker into different networks. And from that model, we calculate the balance of activation across the networks, and we look at how that balance shifts. In young folks, that balance tends to be more weighted towards the control network. And then as you get older, it gets weighted more towards that effort network where you're just kind of scrambling and responding to deal with the noise that you haven't been able to effectively filter out. How that balance shifts in older folks is how it looks in younger folks with the high levels of blast exposure historically.
Well, I think that's a great finding because that's also consistent with some of our research. When we're looking at our veterans with PTSD, the blast TBI is one of the leading factor, and increase the likelihood develop or potentiate the development of PTSD. And that region over there, the medial frontal region, and as part of the top-down modulation, suppress the fear from amygdala, those fear network. So the finding actually is very interesting because that may highly consistent with our MEG findings on PTSD. We see that reduction of activity from ventral medial prefrontal cortex, and meantime we see this increase in the activity in amygdala and hippocampus. So that's finding actually is very exciting. Yeah.
Absolutely. Almost certainly not specific to speech. This is a domain general region. We specifically recruit veterans who report hearing difficulties, and so we're likely to see it in that particular domain. But absolutely, very well, this pattern could extend to other domains requiring that kind of top-down control.
Great. Yeah. Thank you so much. Any other questions? No. Thank you.
Thanks.
Any other people from the audience want to give a presentation?
Yeah, I'll talk a little bit.
Yes. Yeah. Great. Yeah.
So I didn't bring any slides, but I don't need them. So I'm Shane Seibart. I'm a professor of electrical and computer engineering at UC Riverside, and I work in the superconducting electronics community. So it's really exciting to see how far the MEG technology has progressed over time. I think it's one of the top 3 successful applications of superconducting electronics. And in the time when I used to work on sensors called squids like this, one of the things that we were working on is, can we make squids, these sensors that they showed in the helmets and so on, can we make them out of ceramic materials that superconduct at higher temperatures? And a lot of the motivation for that is one, it'll save you money, right? You don't have to cool it down with helium, but now you have this system that recovers your helium and kind of cools it down. So you're just kind of running electricity through it, which is a little bit cheaper. But there was another motivation at the time that we used to talk about, and that is when you move to the high temperature materials, you can relax those cooling requirements and you can bring that sensor closer to the brain. So if I recall properly, you can correct me if I'm wrong, but I believe the niobium low temperature superconducting squids are about 2 to 5 centimeters away from the brain. And then you have, another centimeter, which is like your skull or so. And it's really hard to get those signals out. So you can think of, if you're trying to get resolution on the sensor, you're limited by that standoff distance, right? If you're trying to pinpoint where some magnetic field is coming, you want to get the sensor closer to the brain, but this cooling, this refrigerant, isn't allowing you to do that. So that was one of the key motivations we made. And then there was a second one where the unit that you saw, it's called a doer. You put these sensors in it, and it's fiberglass, and it doesn't fit everybody's head. You try to make it like the average head or whatever, and that'll also cause some standoff of the sensors. And it's harder to cool things with helium than nitrogen again. So we were looking at technologies where we would do like an adjustable sensors in a flexible way out of these high temperature materials and get the sensor as close to the skull as you possibly could. And I think a lot of people worked on that, but NIH has kind of like shifted towards optical sensors and other technologies, and I don't know if people really work on this anymore. But from the superconducting electronics side, the high temperature materials are making progress in other applications like digital electronics, amplifiers, and other sensors. So when I see your presentation, I wonder, so I'm now going to throw a question back to Dr. Lee and Huang. If we could make those sensors smaller and closer to the brain, what can you do with them? Is there some questions that you guys have that, having more resolution and more data would help you answer? So basically, it was just a long question, but that's kind of my contribution. I wanted to talk about the sensors and where electrical engineers can help.
Thank you, Shane. I think that's excellent, and the topic, and so the high TC and the squids, and as you mentioned, have certain advantage. Close to the source, potentially better signal noise ratio because it's so close to the source, and also you mentioned that unlike the helmet, was one size fits all, and the high TC squids can be modular, like a EEG electrode, you can put it anywhere you want, and that actually working towards a development of more like a portable and MEG sensor array. And of course, you mentioned that many of the new MEG development has put the focus on the OPM, the optical pumping magnetometer, but the current OPM have limited and dynamic range. So to make the OPM, we have 12 OPM sensor in our lab, but when that worked very well in our old lab, which you were there before, because the old MEG we have, which is a very simple Nulling coil, we can make the OPM functioning. The reason we need the Nulling coil because the OPM can only be functioning with limited dynamic range. You had to basically cancel out the remaining earth field. But in the old lab, with very simple Nulling coil, we can make the OPM working. But for a new one, because we have a helium recycler that's co-head on top of the MEG array, that big piece of metal generate inhomogeneity, and the perturb the magnetic field, within the shielding room. And so the shielding room would be useless because we are within the shielding room, So, but because the new co-head added over there, all the OPM just fail. And we had to redesign maybe a very sophisticated Nulling coil to make the OPM working. I wonder maybe the high TC squares with maybe a higher dynamic range can overcome those problems, potentially make the MEG recording more portable.
I think that's one place, but also when I think about the portable, like, how important is your room? Because that's not very portable, right? Like, are there some measurements that are useful that you can do outside of the room, or are you really reliant on the shield?
Yeah, that's a good point. So far, all the good high-quality MEG recording is performed inside the magnetic shielding room, but there's a few company, and Twin Leaf is one of them. They're developing. They claim they can develop this MEG sensor. They have one paper published in the open field without shielding. They showed of course multiple repetitions auditory stimuli. They can detect the human auditory response on the unshielded environment. But that's only one example. I don't know how feasible that one's in the noisy hospital environment. I think you still need maybe some simple shielding, and with some of the OPMs, But I think there's still a room for the high TC squares to make big contribution to the MEG recording.
Absolutely. So the closer you get, the better. And so absolutely, if we can get even a couple centimeters closer, that would be a huge advance, because that will really increase the signal, because that's actually the competition. The OPMs, their intrinsic signal noise is not as good as a squid, but because you can get them closer, it actually is competitive. But if we can have the same squids and get closer, that's even better. So that's the first thing. If you can get really close to the thing, that's huge. The second point is it really doesn't matter if it's portable or not. As a physician and stuff, there's a lot of things that aren't portable. You have to go in the hospital and you have to get your MRI. That's not portable. Of course, now they're trying to make portable low-field MRIs, but it's okay if it's not portable. It's not something you need all the time to make a diagnosis of epilepsy or precursor mapping. It's okay. I mean, yeah, would like to do it in your kitchen, yeah, but it's okay. You go to the hospital because that's where you get the best noise, and it doesn't matter if it's portable or not. You can go and drive to get an MRI scan, and it doesn't have to be portable in the wilderness. The question I have is, so do you have high TC squids that you've made that you could be used, and how close can you get?
Yeah, well, maybe I should go back to a microphone somewhere.
Because that would be huge.
Yeah, so I think what's really holding back the high TC squid is the lack of wire. So in a niobium squid, you can make an input coil. So you put a transformer in front of the sensor to amplify the magnetic field and focus it down into the sensor. For high TC, it's a ceramic. So you can't really make wires out of it, but you can make chip transformers. But closing the loop on the transformer, you have to make another ceramic contact to it. So it's just some engineering challenge. And we know how to make that contact. We recently just did a paper on that. So we think we can make, a very small sensor that probably doesn't have the same noise properties as like a niobium because it's shrunken. Where a niobium sensor has like a 2 centimeter coil on the end of it. Our coil would be down to like 1 centimeter or half a millimeter or so, or sorry, half a centimeter. But the noise properties would be a little higher, but if you can get it a little bit closer, I think we have the signal to noise.
Absolutely. I mean, don't give up on that, because that's the advantage. If you get closer, that gives you a huge gain in signal.
Yeah.
And then you can see deeper structures and so forth.
And
what are the other two?
There is a
indications?
guy that can match the niobium squid, and he's retired from squids. Now he does something else in the community. I think Quantum or something. But basically what he would do is he'd make these transformers on a chip, and he'd make about 30 of them. And then he would make 30 squids, and then he would test all of them and find the one out of the 30 that actually got the good noise properties, and then he chip them together. Because if you just try to make them integrated, the probability is 1 over 30 times 1 over 30. You have to make, a lot more of them to get that value. So I think in the community, there's how do you get the noise down of the materials, and we're starting to understand the materials better and how to fabricate them. So I think it's not far off, but I just kind of was wondering where, the OPMs are compared to the squids, because it's NIH, if they just put a little bit of money back into squids, it would help, but they don't fund it anymore. They really, it's all, room temperature stuff that they want.
What are the other uses? You said it's one, MEG is one of the good uses for these squids. What are the other ones?
There's geo surveying. So
yeah.
they use squid sensors, and they put like a big coil on the ground, and they send a pulse down into the ground, and it temporarily magnetizes everything in the ground. And they just use squids to read out the mapping, and they can tell you, oh, there's some oil over there and natural gas there, and they can really find all kinds of resources and stuff underground. And I think that's the last big thing of high TC isn't even squids, but it's filters. So you can make very, very sharp analog filters where we call it a brick wall filter. And essentially you can broadcast at higher, frequencies because you're not polluting the other bands. And they're mostly used by the military now because they were for cell phones, but the cell phone people just figured out, well, if we just put in more towers, we can compensate for it. We don't have to buy this expensive cryo cooler. But for the Navy or so on, they want to block their own signal from their own sensors. So they use these superconducting sensors.
Thanks.
Yeah. Yeah, thanks.
Any questions from the audience?
Yeah, actually, maybe I have a question.
Yes.
So it seems to me that MEG has a one big fault, that it's depending on the fact that only one area in the brain is producing the signal, right? But how do we know that only one brain area is producing the signal? It's not like more dispersed.
Oh, so in certain cases, there's only one area is activated. For example, in the case of a patient with epilepsy, the interactive spike, when there's no propagation, you can treat the one as one source, But, in the example I show, when we localize the human language network, and the test is like a visual stimuli, right? You see there's a baby, apple, whatever, crocodile. So you activated all this, your visual network, then you name that picture, with the language production. You're going to activate it in your, on Wernicke's area, Broca's area. So in that image, if you look closely, there can be a dozen sources light up. So you need to solve the MEG inverse problem, and we use the package that I developed. I'm the main developer called FastVestel, which can handle many, many sources. And it's called MEG source imaging. So back to your question, in some cases, there's only one source. You can treat that one as maybe just one equivalent electric current dipole. But in reality, in the case of language, you have tons, at least a dozen areas light up. You need to solve the problem with a more sophisticated model. And the FastVestel, actually the core is the spatial temporal L1 minimum norm. And give us a sparse solution, but taking into consideration the high resolution in time and space. There's other approaches called, beamforming and also L2 minimum norm, in addition to the multiple dipole fitting. So that's a whole category of MEG source modeling and try to solve the MEG inverse problem. And the more sources you have, the more challenging situation you end up, But that's why I spent about 50% of my time develop or refine and the MEG source imaging technique.
Yeah, so just historically, yes, for epilepsy, and of course, you imagine one simple delta wave function is good for epilepsy. But no, in fact, the history of this solving the inverse problem, Ming Xiang's a major leader. So after he got, so everybody can do single dipole. But then one of the Ming Xiang's first things, now 20 years ago, is a multi-start. He had n, where n can be 2 to 10 to 20 separate sources, and then you can do a calculation for that as well. So punctate single dipole sources, but 20 of them. And he was able to successfully show that you can actually do that quite well. But then the final one, of course, is distributed sources. And that's the L1 minimum norm, and that's fast-Festol. So yes, although the earliest days was with one dipole, and that's still successful for many things. Median nerve stimulation, for example, same thing. It's stimulating the median nerve. It's going to primarily, at one point in time, it's going to be the somatosensory cortex, and then other ones will come at multiple times. But yeah, so there actually means simultaneous, multi-start, multiple dipoles, which Ming Xiang did, and finally, diffuse distributed source. So it's not just single dipole. That's the one I talk about. But Ming Xiang has solved it both for multiple simultaneous dipoles as well as distributed source, not non-dipole.
So my follow-up question would be, so when you compare it to fMRI, right? In fMRI, you can easily see the multi-sources.
Yeah.
How accurate it is, like your multi-sources algorithm?
So in the cortical level, the spatial resolution, spatial accuracy for MEG will be similar to fMRI. We talk about a 2 to 3 millimeter accuracy and spatial resolution using our spatial temporal L1 minimum norm. If you use beamforming or other approach, L2 minimum norm, that resolution for the MEG data will be not as good as our package. But the intrinsic spatial resolution in the cortical level for MEG and fMRI are similar. And of course, MEG have a huge advantage in the 1 millisecond temporal resolution compared with fMRI, which is about 3 to 5 seconds. So in the subcortical level, and the MEG signal, like EEG signal, goes down 1 over R squared. R is the distance between the source and the sensor. So when you go deeper and deeper, the resolution of MEG or EEG, going down. And but with our good shielded ROM, we can still detect reliably the signal from a lot of subcortical neurons, like the one from amygdala, from hippocampus, from insular cortex. Those regions, we routinely see the signal from there. And the one challenging part that MEG cannot reliably detect, but fMRI is very good, is the thalamus. Thalamus. So for thalamus, activity, fMRI pretty good. MEG or EEG, even the cortical EEG, invasive one, is not going to be very good. A couple of reasons. Number one, the thalamus is very deep, really the center of the brain, and far away from the sensor. Number two is, you call my MEG slide 101, we needed the pyramidal cell to line up in parallel to allow the signal submission. That's actually, a good condition for cortical neurons, but for thalamus, the neurons look like a spaghetti, So the signal summation condition is not as good. That's why for MEG or EEG, we're insensitive to signal from thalamus.
But
So
insula. So let's say you have like the temporal lobe, and then you have the insula underneath it. Can you tell the difference?
oh yes, we can. The
Easily.
different depths, and so we can tell the difference between insular cortex and the auditory response. Pretty straightforward.
But there's another thing, in addition to you thinking of activation patch of the brain, the other thing that MEG has that fMRI can't do is looking at the frequency, the native frequencies of the brain. So the brain is operating at, as you showed, multiple frequencies. There's the alpha waves here, but you have delta waves, but beta, gamma, especially gamma too. Because we have instant millisecond temporal resolution and there's no distortion by the skull, we can see all the frequency bands of the brain at all times. And of course, you can do connectivity. So because the fMRI looks at the blood flow to that, you don't have that instantaneous frequency response. So you can do connectivity studies and so forth in resting state, but there's a whole parameter space of the native frequencies of the brain that's a parameter that fMRI can't explore. And that's really important. That's like our slow wave, or the gamma wave activity, all these other things. And then the connectivity between these different frequency bands that fMRI has, that MEG has that parameter space of the intrinsic frequencies in real time, in millisecond resolution, that fMRI can't do that.
Okay, so I think we're perfect timing. I think we need to stop over here and ready for the next section. Thank you all so much.